Okay. Well, welcome to Crime Rounds. It's a genuine pleasure to introduce Ziva. With the changing laws of cannabis, there's been a much more interest in society with cannabis actions, the psychoactive actions of THC and the kind of murky ones of CBD. But I think maybe Ziva will get into that a little bit. It's kind of led to the birth of the Cannabis Initiative in the Semel, which was begun by Peter, Jeff, Jen, Tim Fong, and myself. But for us all, really, maybe except for Jeff, cannabis was our secondary interest and really wasn't our knowledge base. But then came Ziva, and she whipped us all in shape, and she really is a knowledgeable person in this area, and has been an active researcher. So Ziva's really shown incredible leadership of the cannabis research at UCLA and matured this initiative into a center which is now really cross-campus and has many different facets to it. So Ziva is the director of the UCLA Center of Cannabis and Cannabinoids in the Semel Institute. And she has appointments both in psychiatry and anesthesiology, and we can't let her go to anesthesiology. So her research has many avenues. She works with the police on intoxication, and I've been at some of those meetings. They've been kind of interesting. She has NIH grants on opioid sparing for pain, other grants on terpenoids and flavonoids to determine the entourage effects, the mixture effects of all these different compounds in the cannabis plant, which is very interesting. So our grant portfolio has really been growing really exponentially. I won't use the analogy of weeds, but I won't. Okay, so Ziva and myself have developed the course together, and we're teaching that now on cannabis and the brain. And this is part of the minor, which is associated with the Semel, and been very popular with undergraduates. So we're actually got a test, a midterm tomorrow on that, which we're busily trying to put together, because we initially were going to have an in-person test, and this in the Semel auditorium, but that got flooded and is out of commission for a while. So a little bit of background about Ziva. She received a PhD in Michigan, the University of Michigan, and with Jim Woods, and he's a very renowned pharmacologist and a good sailor as well. Then she went to Columbia, where she's working with people, getting to work with people, and Sandy Connor and the crew there, and she started really doing some clinical research there on cannabis. And we recruited Ziva sort of during 2019, and though there was much coaxing involved, she eventually came, and we're so happy that she did. Ziva's achievements have been many, but I'll include some here. She was, I think, really important was she was on this influential National Academies of Science Committee, which really has been very important in sort of setting the sort of field straight about the effects of cannabis, what's authentic or what's not. And that's been very important. She's president-elect of the International Cannabinoid Research Society, past board director of the College on Problems of Drugs and Dependence, associate editor of the Journal on Drug and Alcohol Abuse, and on many other editorial boards. In my view, Ziva's a rising star in our department. Very happy that she came, and privileged to be part of the Cannabis Center. So Ziva, take it away.
Chris, thank you so much for that wonderful introduction. And everybody knows how happy I am that I am here as well. I mean, I'm always walking around with a big smile on my face, so it's great to be here. And I'm so excited to talk to everybody today. Wish that it was in person, but so it goes. So today I'll be really focusing on probably a semester's worth of cannabis and cannabinoid research and discovery, as well as what happened here at UCLA. So hoping I can get to everything, and let me just, okay, so hopefully you can see. Okay, Ernie, you'll let me know if you can't see it. Okay, so today I'll be talking about 50 years of cannabis research at UCLA, although we won't really be getting to that until the very end. But I'm going to talk through the history of cannabis public policy and discovery with respect to cannabinoid pharmacology. And I'm going to be emphasizing along the way the work that we're doing here at UCLA that is really helping to further the field. For disclosures, I have funding from a variety of federal and state agencies, and I've also consulted with Canopy Growth Corporation. I received study medication from the NIH, as well as Canopy, and I received drug administration supplies from a company called Storrs Bickel. So a brief outline of what we'll be talking about today is the history of public policy and pharmacology of cannabis and cannabinoids. I'll then talk about what is known about the therapeutic effects, what is known about the risks of cannabis use, how UCLA is furthering knowledge in both of these domains. And then again, at the end, I'll talk a little bit about UCLA history of cannabis research that it's actually been a lot of fun to learn about over the last couple of days as I was preparing for this talk. So we're not going to start back in 2000 BCE when, the first remnants of cannabis were discovered. We're going to start off in the United States in 1932 with the Marijuana Tax Act, which was essentially the first ruling that restricted the use of cannabis. And this was specifically for non-medical purposes. In 1942, cannabis was removed from the pharmacopoeia. So even though it wasn't regulated, medical cannabis at that point, it was removed. But there's a rich history of the use of cannabis for medical purposes. Of course, around the globe, that and you'll hear about this in all talks, that the history of cannabis use for medical indications has been a long one, but also here in the United States. Now, during this period of time, there was a great deal of interest in understanding the different constituents of the cannabis plant. And at that time, there were researchers, both here, organic chemists here and abroad, that were really trying to understand the chemical constituents of the cannabis plant. And this was actually the first time that there were chemicals in the cannabis plant that were isolated and that the structures were elucidated. So before we knew about delta-9 THC, CBD, cannabidiol, was really the first phytocannabinoid that was isolated, and cannabinol was actually isolated back in the 1800s. Cannabinol is also a constituent of the cannabis plant, and its structure was elucidated in 1940. Then came along the isolation and structure elucidation of delta-9 THC, the cannabinoid that I think we probably know most about at this point in time. And this was really instrumental in setting the groundwork for understanding cannabis and cannabinoid pharmacology. In 1964, this happened with Yechiel Gaoni and Rafi Meshulam in Israel. And they not only isolated delta-9 THC and elucidated its structure, but they also were able to determine that delta-9 THC is, in fact, the psychoactive and intoxicating component of the plant. And these studies were done across species. And so this discovery really set off decades worth of research looking at delta-9 THC's effects across species with respect to understanding its potential therapeutic effects and adverse effects. With that discovery came along again, decades worth of research into this plant. And here today, we now know that there are over 140 phytocannabinoids in the cannabis plant. We also know that there are other chemical constituents that we'll talk about in a bit called terpenes and flavonoids in the cannabis plant. They're not necessarily unique to the cannabis plant, but they are thought to have biological and psychoactive activity. At this time, the NIMH contracted the University of Mississippi to be the first grower of cannabis in the United States. And you'll learn that it stayed this way for a very long period of time. And this was really important. Having a supply of cannabis that was grown, it was standardized, that was used specifically for research, really helped to pave the way for research on the cannabis plant. And in fact, this is the cannabis that we still use today. And here's a photo of what this plot of land looked like back in 1968. And here's Mahmoud El Soli. If those of you who have heard talks about this farm have surely heard from El Soli, who came as a postdoctoral research fellow in 1975 and has been there since. So they moved from having just an outdoor facility to a very controlled indoor grow, which is what you see here. Next, the next landmark issue was the Controlled Substances Act that Nixon commissioned, which essentially put cannabis as a Schedule I drug. And this essentially deemed cannabis as having no therapeutic effects. And therefore, not only was non-medical use and possession illegal, like it was in 1937 with the Marijuana Tax Act, but so was medical use. So medical use was prohibited with the CSA. And here you can see that marijuana and tetrahydrocannabinols are put in this Schedule I category. And the Drug Enforcement Administration was set to lead the enforcement of this in 1973. And what that meant was that anybody who wanted to study cannabis or tetrahydrocannabinols was required to have a Schedule I registration. Now, we'll talk a little bit about how the restrictions were a little different back in the 1970s versus now, but for right now, this Schedule I scheduling, the Schedule I categorization of marijuana has led to one of the primary barriers of research of cannabis. For example, in order to obtain a license, it requires quite a deal of time and money. It also requires federal and state approvals. The institution has to provide support and also has to comply with the requirements that are needed for this drug storage. And these drug storage restrictions really limit the type of study design that people can pursue with respect to their cannabis research. And so here's just a snapshot of what's entailed when you obtain a license. So you have to have security, which can cost up to $3,000 and monthly fees. You have to have a double lock storage facility and a very large safe. So somebody who's trying to get in this field has to already have the resources available to be able to build out their storage facility. So what this also means is that not only is it difficult for researchers to obtain a license, but it's also difficult for people that are manufacturing the study drug to also get a license. And so what ends up happening is that there are very few available sources for a study drug, which impacts the research. So we had the DEA scheduling of cannabis happening in 1970 to 1973. But interestingly, in 1985, what ended up happening was because there was a great deal of research and interest in Delta-9 THC, there were a number of studies looking at its potential therapeutic effects, specifically for nausea and vomiting and appetite increasing effects of Delta-9 THC in patients with cancer. And so in 1985, the FDA actually approved the use of synthetic THC called Marinol, as well as Nabilone, a chemical that's very similar to Delta-9 THC. And essentially, when the FDA approved the use of these medications, the DEA had to reschedule Delta-9 tetrahydrocannabinol in these forms specifically. And so this demonstrated that at least for this component of the cannabis plant, there are approved therapeutic uses. Next, we had a significant discovery in cannabinoid pharmacology, which is that the CB1 receptor, the cannabinoid receptor that THC binds to, was discovered, cloned, and localized. And this was a paper in PNAS demonstrating across species through autoradiography where the cannabinoid receptor was found. At that point in time, it was just the cannabinoid receptor, it wasn't CB1. And what these researchers found was that there was dense expression of this receptor in areas that controlled feeding, memory, movement, reward, all areas of the brain that we think of as being implicated in THC's effects. What's interesting, though, is that the CB cannabinoid receptor was not densely located in the pre-Pottinger complex in the brainstem. This area of the brainstem is thought to be involved in regulating respiration. And so frequently you'll hear that there's no way that you can overdose, there's no lethal toxic dose of THC, because essentially there aren't cannabinoid receptors in that area of the brain that controls respiration. What this study also found was that there was conserved distribution across species. And this finding also led to the acknowledgement that the CB1 receptor is actually the most abundant G protein coupled receptor in the brain. So more abundant than dopamine receptors, serotonin receptors, and other G protein coupled receptors. So this was a really significant finding. And that finding led to other elucidations of the endocannabinoid system, such as the CB2 receptor. The CB2 receptor was discovered, and then the endocannabinoids were discovered, anandamide and 2-AG. And those discoveries helped us to understand where there were CB1 receptors in the brain and body, and where there were CB2 receptors, primarily in the periphery, in the brain when there's a pathological state, and why these two receptors were very important. What did they control? We also learned about these two endocannabinoids, anandamide and 2-arachidonyl glycerol, 2-AG. With these findings in the endocannabinoid system, we learned about how the endocannabinoid system is helpful in maintaining homeostasis. At that point in time, in 1996, came the landmark change with the California Compassionate Use Act. So California was the first state to essentially legalize cannabis for medical purposes. So this was a huge development. What medical indications were approved? So we have these 12 indications, and the last indication is really other chronic or persistent medical symptoms. So really a catch-all. So with this change in legislation came a series of questions that were on people's mind. Is cannabis therapeutic for these approved indications? If so, how can it be used, and should it be used? What are the risks associated with use? And so I'm going to talk a little bit about what we know about these central questions in a minute. Next, we learned on the discovery side how endocannabinoids worked, and this was through retrograde signaling. So unlike other neurotransmitters, endocannabinoids act with retrograde transmission, and essentially they work at glutamate and at GABA receptors to dampen those signals. And so that was also a very important landmark finding. Around 2012, Colorado became the first state to legalize non-medical cannabis use. So some people refer to this as adult use cannabis, other people refer to this as recreational cannabis. So that was a significant change, and California wasn't far behind. In 2016, Proposition 64 was passed, essentially legalizing adult use cannabis. And in 2018, this was implemented. So dispensaries started opening up. I don't know if you remember, but January 1st, 2018, the Hollywood sign was changed to the Hollyweed sign. So people, of course, were very excited about this. And there were a whole slew of states that really followed. And here you can see the states in blue that were legalizing cannabis for adult use or recreational use, and the states in red that were legalizing for medical use. So in 2017, there were 29 states plus DC that legalized for medical, and 8 states plus DC that legalized for recreational. And this change in legalization led to a whole host of questions. Will greater access for both medical and recreational use impact use? Will this have potential negative consequences? How do you regulate this market? And what are some of the unknowns that we have to think about and get ahead of before we get greeted by them? In the same year, Epidiolex, which is cannabidiol from the plant, was approved. And this was really important because it was the first time in decades that the FDA approved a cannabinoid for some medical indication. It was also the first time that the FDA approved a plant-based medication, a cannabis-based medication for a medical indication. And because of this, CBD at this point in time, it was still Schedule 1 at the federal level. With this movement, with the FDA approval, the DEA rescheduled Epidiolex to Schedule 5, and around a year later, it actually went off of the CSA, so it was no longer scheduled. This is the same year that the 2018 Farm Bill was passed. This is a federal bill essentially legalizing the cannabis plant, a specific cannabis plant, hemp. Hemp is a cannabis plant with very low levels of delta-9 THC. But what that meant was that now cannabis, hemp, could be grown across the United States, and this hemp can be used for fiber and for structures and used for other things that we don't usually think about cannabis being used for, but it also could be used to grow and be a source of cannabidiol and other types of cannabinoids. So this was a really important movement. And then lastly, in 2021, remember that in 1968, we had our first cannabis farm. It wasn't until 2021 that we had additional DEA approved cannabis farms. And so this was significant because essentially what this did was this opened up the ability to get study medication cannabis from other sources. Now, I will say that even though the DEA has approved these cultivators, we don't have product from these cultivators yet. So this is where we are in 2022. In 2022, even though cannabis is federally illegal, we have 17 states plus Washington, D.C. that have legalized it for recreational purposes or adult use purposes, 37 states for medical use plus Washington, D.C., 11 that have state laws related to CBD. 3 states have been holding out at this point, which they really don't have other cannabis laws. There are 5 other states that are on the ballot for this year to legalize for adult use, non-medical use. So with these changes, we had those questions we had before: will greater access impact use? What are the potential negative consequences? What are the unknowns on the horizon? And here we've been learning about the unknowns on the horizon. And this was the industry and emerging trends of use. And so let's talk a little bit about these emerging trends because they have significant public health impact. We hypothesize at least they do. So one important trend is the increase in strength of cannabis over the last several decades. So this is a graphical representation of how THC percent, this is how we define cannabis strength, how it's changed over the years. And this is seized cannabis. So this is not the cannabis that you get in dispensaries. What you can see here in 1995, the average percent of THC in cannabis that was seized was 4%. In 2014, it went up to 12%. If you carry this chart out until 2017, it was up to 16%. If you go into dispensaries now in California, the Westwood Pharmacy, you will see cannabis that has up to 30% THC or even 35% THC. So we know that cannabis strength is increasing. What is the impact of these, the rising cannabis strength on a number of different variables? So we have Mike Roth here in pulmonology, who has a grant to look at how does cannabis potency impact secondhand smoke. So this is a very exciting area of research. Not much has been done with respect to secondhand smoke and cannabis smoking. We also have changes in modes of use. So people are no longer just smoking cannabis. People are vaporizing it using these different apparatuses. So we have a vaporizer and vape pens. People are using edibles more frequently. People are using topical products. So people are using cannabis across a range of modes of administration. We also have the emerging trend of high potency products that are almost 100% THC. So this is a far cry from plant cannabis that people grew up with in the 1970s, 1980s, 1990s. This is essentially almost pure THC that is used as through inhaling. So we have a grant here to understand, should these high potency products be taxed? So should we implement higher taxes on higher strength products? If we do, would that potentially nudge consumers to the lower strength, potentially lower risk products? So this is a very exciting area of research that we're involved with. We also have CBD products that are emerging. We have CBD products that can be used as oral tinctures, that can be used as pills. We have wearable clothes that have CBD infused in them, pillowcases that are infused with CBD that promise you a restful night's sleep. We have other cannabinoids that, that manufacturers are starting to market. So cannabinoids such as CBN that promises a restful night's sleep, CBG, cannabigerol, CBXYZ, across a range of administration through vaping, tinctures, oil, salves, and others. We also have this rising interest in the terpenes. I talked a little bit about terpenes, how terpenes are in the cannabis plant, and they are also thought to have certain maybe medicinal properties to them. And so when you go into dispensaries, frequently you will see products that tout that they have a certain number, a certain concentration of specific terpenes in them. So the question is, what are the effects of these products? Are they safe? Are they therapeutic? So people are using these products thinking that they might be helpful for a range of conditions. So we actually don't have a good handle on if these products are safe or therapeutic at this point in time. There's very little in the literature apart from survey studies. There are some preclinical research in this area, and we have one researcher here, Kathy Kahl, who's trying to understand some of these terpenes as novel analgesics in a preclinical study. We also have a couple of human studies looking at these terpenes, specifically myrcene and beta-caryophyllene, as well as one of these cannabinoids, cannabigerol, to understand their potential therapeutic effects, as well as adverse effects. Essentially, these are the first studies that are looking at these chemicals under controlled situations in humans. So this is really important area of research to help inform people about what it is they're taking, what are some of the risks that they should be aware of, and if there are potential therapeutic effects, could those products or could those terpenes and cannabinoids be moved into a study with a patient population? So how do these changes in legalization impact cannabis use patterns? So cannabis use is increasing, okay? So this shouldn't come as a surprise to many people. In fact, when you look at past year cannabis use, you can see that among the 18 to 25-year-olds, this is just past use, we're seeing a slight increase since 2002. What's reassuring here is that when you look at past year use and you look at the 12 to 17-year-olds in the gray line, we're actually not seeing an increase. In fact, we're actually seeing a slight decrease. And this was contrary to what a lot of people thought would happen when cannabis became legalized in these states. So this is comforting. Now, the question is it possible that there might be certain threats to this decreases in cannabis use in 12 to 17-year-olds? For example, the marketing that's being used to target perhaps this younger cohort. And so we have a researcher here, Dara Garamani, who's actually looking at this. He's interested in understanding how cannabis marketing in California youth might push people to desire, to purchase, to use these products. A recent finding just came out from Monitoring the Future that looked at past year cannabis use among 19 to 30-year-olds. And what they found, again, what I just showed, was that the highest rates of use essentially are between 18 to 25-year-olds. Monitoring the Future looked at this specific age range. And what they found was that between 2011 and 2021, there was a 13% increase in annual use. Now, those of you who have heard me talk about this before, do we really care so much about annual use? If people are using it once in the past year, is that really a public health concern? For me, I think that probably if they're using it monthly, weekly, daily, those are really the important statistics. And so if you look at this population and you look at their past month use, what you see is that these annual users are actually predominantly using at least in the past month. So we saw in 2018, 17% of this cohort using cannabis in the past month. In 2021, you can see that it almost doubled there as well. And then if you look at daily use, you're also seeing a significant increase from 6% to 11%. So what are the outcomes with respect to this increase in use? So we have Howard Padwa and Darren Urata who are assessing the impact on legalization, specifically in the state of California, Proposition 64, on how Proposition 64 is associated with cannabis use, maladaptive cannabis use disorder treatment, and public health. And so these findings will really be instrumental in us understanding how the passing of adult use cannabis laws impact cannabis use patterns and public health outcomes. So we know that cannabis use is increasing across the board in specific age categories. So let's talk a little bit about who is using cannabis now. Has there been a shift in the type of people who are using cannabis? So the shifting demographics of the cannabis user. And so one area of research that we focus on at UCLA is specifically cannabis use in women. And part of the reason why we focus on this, as will soon become clear, is because we're starting to see that although in the past, women were thought to be protected from cannabis use. So across surveys, men always outweighed women 2 to 1 who were using cannabis. We're also starting to see that there are certain cannabis products that are being marketed specifically to women and for women's health conditions. And so here's an example of how cannabis use is increasing among women. Again, here it's just 18 to 25-year-olds. We have males on the left here and females on the right. And you can see between the years of 2015 and 2018, monthly use pretty much stayed steady among the males. But among the females, we started to see a significant increase. And if you look at daily use, you see also that there's been a significant increase in females in daily use over that 4-year period, as well as cannabis use disorder. So we're seeing increased use among females, and we're also seeing increases in cannabis use disorder. And this is not just restricted to the 18 to 25-year-old age range. We also know that this is happening among women who are 26 and older, where there was an increase, a 40% increase in both monthly and daily use during that time period. We also know that in addition to non-medical cannabis use, there is a growing female demographic of medical cannabis use, specifically for pain and some other conditions, but pain is the primary indication for which people are using cannabis. In fact, in many surveys, 50% of the people surveyed who are using medical cannabis for pain are female. So the question here is, does cannabis, specifically THC in the cannabis plant, does it affect males and females differently on a whole range of outcomes? Specifically, what we're interested in here are abuse liability. Do we have to think about how the abuse liability and the transition from using cannabis to developing a cannabis use disorder, does that differ between males and females? And since it seems like females are using cannabis for pain relief, are there sex-dependent effects with respect to the analgesic effects of cannabis? And if so, what are some of the underlying biological variables that might underlie these differences between males and females? So here are a couple. Do males and females differ in their pharmacokinetics of THC? Do they metabolize THC differently? Are there differences because of some hormonal modulation of THC's effects? Is it possible that females or males might develop tolerance to these effects faster than the other sex? And I'm choosing these variables specifically because if you look at preclinical literature, there are significant differences between males and females with respect to their response to THC. And these are 3 variables that impact the sex-dependent effects. And so we have a grant that is exactly looking at this effect, comparing males and females with respect to THC's abuse liability and analgesic effects, and probing the variables that underlie these differences. The next demographic that I think we're really interested in happens to be the aging population. And so some of you might have seen this article in NPR, and there's an article in the New Yorker about how there are buses coming to senior living facilities, and they're taking the people that live there to these dispensaries. They're having bingo at these dispensaries, and they're trying to educate the older population on what cannabis is and how it can be used. So has cannabis in the aging population increased over time? And this is a snapshot of, you'll have to excuse that I'm using this from the Washington Post, but it actually demonstrates really nicely how over the years, the population of 55 to 64-year-olds, this is a nationally representative sample, their monthly use in 2017 was on par with 12 to 17-year-olds. So here are the 55 to 64-year-olds. You can see that there's been an increase, and the 65 plus, there's also been an increase. So this is a considerable increase in use in these populations. In fact, past month use in people from 60 to 64 years old doubled between 2013 and 2019. And if you ask people why they're using it, especially people who are using it for medical purposes, what you'll find is that 50% of people 50 and over are using cannabis specifically for pain. So that led us in the lab to think about, well, if older people are using cannabis for pain or for other indications, do the effects of cannabis differ between that population, the aging population, and a younger population? And when you dig down in the literature, there actually hasn't been controlled studies addressing this issue. Most of the studies that have looked at cannabis effects in human labs are really looking at people between the ages of 21 and maybe 40. Very few are looking at older populations, let's say 55 to 65, and comparing the effects in younger populations, maybe middle age, 35 to 45, and emerging adults, 18 to 25-year-olds. And when we look deep into the existing data that we had to try and answer this question, we tried to look to see in our own existing data, do older participants differ in their response to cannabis with THC than younger participants? And here is a figure where we administered smoked cannabis, either placebo in the open symbol, so that's cannabis with no THC, versus cannabis with 4% THC in the closed symbols. And what we did was we asked people after they smoked the cannabis, please tell us, rate the good effect that you had from this cannabis. And what we found was that in the yellow circles, the older participants actually didn't have as robust of a response as our younger participants. And same thing with intoxication. The older participants didn't seem to get as intoxicated as the younger participants. Now, some of you might be asking, well, were these older participants tolerant to cannabis? Were they using more outside of the laboratory? And for this tiny little bit of data here, we made sure to control for cannabis use in these populations. So consistently, both groups were daily cannabis users. So this doesn't seem to be a tolerance effect here. So some questions come to mind. Are there differences with respect to other measures of abuse liability, right? We want to understand, are younger people at risk for the addictive-like properties of cannabis at a higher rate than older participants? What about cognitive effects? Certainly, if you're looking at an older population, this is a really important question to ask. Is it possible that the older population might also be less sensitive to the cognitive impairing effects than the younger population, which would be great, but we have yet to answer that question. What about analgesic effects? If 50% of the people who are using cannabis for medical purposes are using it for pain in that older population, it's important to understand, are there differences in the analgesic effects of cannabis? What might underlie some of these differences? Is it possible that there's pharmacokinetic differences? And so we have a grant to actually probe this question. So we're really excited about that. How about the impact of legalization in California specifically? So I touched on a couple of grants that are focusing specifically on that. And we have some other ones that are specifically interested in understanding the public health disparities and the equity impact of Proposition 64, this passing of adult use cannabis in the state of California. And I'd like to call your attention to one in particular that is specifically interested in understanding the employment conditions and equity in the state of California for people that are in the cannabis industry. And so we're really excited to see the outcomes of these studies, and you can learn more here with that QR code. So I promised that I would give you a synopsis of what we know about cannabis and cannabinoids as effective therapeutics. And here I will be pulling on the National Academies of Sciences report that Dr. Evans referenced during his introduction. And so for that report, we were interested in understanding what is the strength of evidence we have supporting the use and utility of cannabis and cannabinoids across medical indications. And for this, we use the evidence from randomized controlled trials, so placebo-controlled trials, and we chose 20 most qualifying conditions across the United States for which people can get medical cannabis. And what we found was that there was substantial evidence supporting the utility of cannabis and cannabinoids for the indications in red: chemotherapy-associated nausea and vomiting, HIV-associated anorexia and cachexia. These indications already have FDA approval for THC, as well as seizure disorder. And I just told you that Epidiolex was FDA approved for specific seizure disorders. And we also found substantial evidence for the utility of cannabis and cannabinoids for chronic pain. Now, this was kind of a catch-all. Chronic pain is a lot of different things. And really at that point in time, there were only 40 randomized controlled trials looking to understand the effects of cannabis and cannabinoids on chronic pain. So even though we concluded that there is substantial evidence, there's still a lot of unknowns. And Veena Ranganath, a rheumatologist here, is doing a study right now trying to assess the impact of cannabidiol, which is thought to have anti-inflammatory properties, on rheumatoid arthritis. Other indications for which there has shown promise for cannabis and cannabinoids include addiction, so substance use disorders, anxiety, PTSD, schizophrenia, and other psychoses. So for addiction and anxiety and schizophrenia, really those studies were centered around cannabidiol, not THC. And we have work in this area as well. Doctors Edie London and Rich De La Garza are currently doing a study trying to understand cannabidiol as an adjunct treatment for participants with opioid use disorder. So to summarize, we have these indications for which there's promise for cannabis and cannabinoids. But one thing that's important to understand is that overwhelmingly, the studies that were done here were not done with cannabis, and they were certainly not done with dispensary products. What were they done with? So largely they were done with these 3 FDA-approved cannabinoids: dronabinol, which is essentially synthetic THC. It comes in a pill form, and it comes in a liquid form. They were done with nabilone, a THC-like synthetic cannabinoid, and Epidiolex, a plant-derived CBD. There were some other studies that were done with Sativex, a drug that's approved in Europe and other countries, which is plant-derived, and it's a oral mucosal spray combining both CBD and THC. And there were a few studies done with inhaled cannabis. A lot of those studies got that cannabis from that cannabis farm. And so this is essentially what that cannabis looks like. We get these cigarettes, they're about 900 milligrams, and participants smoke the cannabis, or they can vaporize the cannabis using these 2 delivery methods. So to conclude, is cannabis and cannabinoids an effective medicine? So based on what we know thus far, the bulk of the research has been done with THC or THC-like drugs. We are now starting to see more research done on cannabidiol, and hopefully with some of those other cannabinoids that I mentioned before. There's 4 FDA-approved cannabinoid-based medications. Only one of them is from the plant. The modes of administration that have been studied thus far, even though you see topicals all over the place, really they have not been studied yet, especially not been studied under placebo-controlled conditions. I can think of only one study that has looked at those topicals. The modes of administration that have been studied here is oral administration as well as inhaled. The effective doses, the doses that have been used in these studies, do not necessarily match what's available on the market in dispensaries. And so that's really important to keep in mind when thinking about how effective and safe are the cannabinoid products that are commercially available right now. So it's clear that legislation and popular belief are really outpacing science, but I think that some of the work that we're doing here at UCLA will help to fill some fundamental gaps. What are the risks associated with cannabis use? So one risk, and actually this conclusion was based off of the work that was done here with Dr. Tashkin and Dr. Roth. One of the risks associated with cannabis use, cannabis smoking in particular, is frequent bronchitis. It was assumed decades ago that one of the risks associated with smoked cannabis was lung cancer. And we know now that there is not an association between smoked cannabis and lung cancer. And that, again, was based off of the work of John Tashkin and Mike Roth. We know that there's a risk of motor vehicle crashes with THC. Maternal use of cannabis has been associated with low birth weight, preterm birth, and admission into the NICU by the neonate. We know that cannabis with THC is associated with impaired learning, memory, and attention. And we know that there are certain mental health issues that we are aware of that are associated with cannabis use, such as schizophrenia, depression, anxiety, suicidal ideation, and cannabis use disorder. And what I want to say here is that for the most part, with respect to the mental health risks and cannabis, we can say that across the board, at this point in time, we know that they are associations, except for one mental health disorder, and that is cannabis use disorder. We know that this is a causal effect. You cannot develop cannabis use disorder without having used cannabis. 20% of users, cannabis users in the past year, will meet criteria for cannabis use disorder, any criteria, mild to severe. 5% of annual users will meet criteria for severe disorder. Now, one of the reasons why, this of course is an issue, but part of the reason why it's a real issue is that some people seek treatment, but right now there are no FDA approved medications for people who have cannabis use disorder to help them stop. So we have one study here. Larissa Mooney is the site lead for a multi-site study looking at a novel medication to help people with cannabis use disorder. So this is really exciting. We're about to get this underway. It'll be great to have a study looking at a pharmacotherapy for cannabis use disorder here. So all this is to say that we have a rapid growth in the industry, lots of different products, new ways to use cannabis. We have many people using, we have new demographics who are now using cannabis for different types of reasons, either adult use, medical use. We know that there is therapeutic benefit of cannabinoids, certainly with delta-9 THC. We're learning more about cannabidiol. We also know that there are adverse effects, but certainly there are a lot of unknowns with respect to the safety and effectiveness of the products that are available in dispensaries. So how are we filling the gaps in knowledge here? So we have our UCLA Center for Cannabis and Cannabinoids, where this is a multidisciplinary initiative that was started in 2017. We have people from across campus who are coming together to be able to tackle these really important questions, to understand the consequences of use, as well as work on our efforts to educate and outreach and do outreach. So we have 90 people from 90 faculty members from 15 divisions and schools, at least 30 people. We have Chris Evans, who's really spearheaded this undergrad and graduate course. We now have, I think, 80 people enrolled. This is our third year doing this. We have our postdocs and our graduate student, Stephanie Lake, Connor Murray, Brittany Henry, helping with this, and Vince Acebo, our administrator, who's helping with this. We have monthly faculty meetings where we have presentations, we have cannabis grand rounds, we have community webinars at an annual event. So it's really exciting to see people here at UCLA gather around this effort. We're also very dedicated to research. Hopefully that's become very clear. Something that I think that we're all proud of is this Cannabis and Cannabinoid Research Laboratory, which is very unique. Not many institutions have a laboratory where the researchers can give cannabis and cannabinoids to participants across modes of administration. So smoked, vaporized, oral. And the premise of the studies that we are doing in the Cannabis and Cannabinoid Research Laboratory are really built off of preclinical evidence, understanding the potential adverse effects and potential therapeutic effects of these cannabis and cannabinoids. We also bring in compelling population data. What are people using? What are people reporting? People are saying, for example, that CBG is really helpful with their sleep, really helpful with their anxiety. Well, let's look at CBG. What doses of CBG? What mode of administration? What's the time course? Also, we always make sure to look if there's existing randomized controlled data that can help navigate the study design. And so this is what our study session room looks like. You'll notice here this kind of weird thing it looks like in a dentist's office, but this is part of our designated exhaust system. So essentially when people smoke or vaporize, the hallway doesn't smell. It's really kind of amazing. The smoke goes up through Edie London's hood right above us on the 4th floor of Semel, and then up through the roofs. So we don't have any kind of stink. We don't have any environmental hazards that are associated with secondhand smoke, for example. And so here we have our cannabis cigarettes here, as well as our cannabis vaporizer. Here you can see Stephanie preparing a dose of THC for vaporization. The THC is aerosolized, it's heated up, and the participant inhales the THC from this bag. And I can tell you more about this bag, why it's opaque. This all looks very unusual and interesting. And I welcome you to come to the lab someday to check it out. So our primary goals is to really help be a resource to enable research across UCLA. Cannabis research is hard. There are so many regulations, and we don't want people to get hung up on those regulations. We don't want people to get hung up on the intricacies, and we're here to help. We're interested in understanding the consequences, both adverse as well as potential therapeutic effects. And investigating how policies governing cannabis availability have impacted behavior and economics. We really place an emphasis on education and research training. We have around 10 undergraduate students in our lab. We have 2 graduate students, 3 postdoctoral fellows. So we're really working on training the next generation of cannabinoid researchers. And also outreach. We place a significant importance on improving cannabis literacy in the community. People walk into dispensaries and they don't know what they're looking at. You know, it's very confusing, and they really have to rely on people who work at those dispensaries to help inform them. And so one thing that we are interested in doing and that we've worked on doing is really helping to improve people's understanding of what is cannabis and cannabinoids. When you see a billboard or when you read a claim in the media, how do you parse that out? Does CBD really help with insomnia? How do we know? So we're really helping to improve cannabis literacy. At least that's my hope, that we're helping to do this. That's our motivation. And so we are not the first ones at UCLA to do this. In fact, UCLA has a really rich cannabis history, and I could spend an entire hour just on that. But I want to point you to a seminal study that happened at UCLA in 1976. It was a 4-year study, so it started exactly 50 years ago. And it was a study, 94-day inpatient study, where Sid Cohen decided that he wanted to understand a range of outcomes with respect to acute cannabis exposure and chronic cannabis exposure. So participants lived here for 94 days, and you can see that they actually earned quite a bit of cash. The study drug was essentially exactly what we study now. It was the cannabis from the farm in Mississippi. And in fact, we're about to start a study looking at cannabis cigarettes with 2.2% Delta-9 THC. Participants got a cannabis cigarette to smoke. The first one was free, and then they had to pay a quarter for every cannabis cigarette afterwards. And you can see that participants self-administered quite a number of cannabis cigarettes while they were here, an average of 5.2 a day. Many outcomes of interest, and a lot of the findings from these studies led to future research, including our future research. At the same time, Sid Cohen was very much involved in understanding the impact of cannabis on the lung. And so he brought Donald Tashkin, again, this pulmonologist, to look at these effects. And so here's a paper that was published in 1973, where Don Tashkin started looking at the acute effects of marijuana, THC, either smoked or orally administered, on lung health. And so from this research program, Don had a R01 from NIDA that lasted 30 years, and he passed it on to Mike Roth. This was an extremely multidisciplinary effort. He worked with epidemiologists, he worked with folks in Semel, he worked with toxicologists. It was really an impressive research program that led to over 100 publications on the effects of cannabis and lung health. Now, some things haven't changed. For example, it took him 2 years to get his DEA registration for cannabis. He still had to go through California state approval. Back then, the RAPC was actually called crack. They changed their name, but it was really hard to get these studies approved. He also had to go through the FDA. There was no IRB at that time and no EH&S. So you saw our session rooms where we had a designated exhaust system. So I talked to Don Tashkin, and I also talked to Evelyn Cedarbaum about what it was like those days. And essentially, Don's lab was in an office space right down the hall from the chair of the school, from the chair of medicine. And people would go visit the chair, and they would smell the cannabis wafting from the office. And Evelyn Cedarbaum, who was here, right here on floor 3 of Semel, she was able to tell exactly when there was somebody smoking cannabis because it would just waft down the hall. There was very little regulation back then. So some things have changed, but really it's full circle. So the institution, UCLA, was really supportive of this research back then, and they are still very supportive of this research now. And I think that's unique. Not all institutions are supportive of this. So I urge you to learn more about what we do. We have a website. We have a listserv. Please email us here if you want to be listed if you want to be added to the listserv. And I'd like to acknowledge some really key people who were fundamental at getting this going, as well as the core people that are associated with the Cannabis and Cannabinoid Research Laboratory, our funding support, as well as the study participants. And I'd like to thank you so much for listening. And with that, I'll be happy to answer questions. I can't hear you, Chris. I think you're still on mute.
Ah, there we go.
There you go.
Yeah, I forgot about that. I thought I demuted, but maybe it didn't work. Okay, so Joaquin Fuster was the first question we have. And again, thanks very much for a very informative talk there, Ziva. He's asking about the role of cannabis of driving. I know you mentioned that a little bit, but you know, maybe you can talk a little bit about the work with the police you have ongoing there, because that's their one of their basic concerns, right?
Right. Yeah. And so at this point in time, you know, it's very difficult. We don't really have a good biological marker to indicate if somebody's used cannabis recently enough that they're impaired and they're intoxicated. And so we're working with the California Highway Patrol to understand if there is a good marker for that. But essentially, if you look at the effects of cannabis under acute administration paradigm, so when somebody smokes, there are effects that impair driving. And in fact, there was a really cool study that happened last year in the Netherlands where they actually allowed people on the road. They had a controlled drug administration study. So they knew if somebody had, you know, somebody smoked a placebo cannabis cigarette versus a cannabis cigarette with 10% THC, and they actually let people on the road. Obviously, there was somebody next to them who had controls in case things went off the mark. But yes, there is indication that acute cannabis, at least smoking, does impair driving capabilities. Now, the question is how long does it impair driving capabilities? And also, what are the doses? What are the modes of administration? Does smoked cannabis have the same impact as oral THC? And so these are some of the things that we're working on now, in addition to understanding what about CBD? People are using CBD because they don't want the THC. The THC is intoxicating, CBD isn't. Well, does CBD potentially impact driving? We don't think it does. But what happens if there's a little bit of THC and CBD? Is there a potential interaction there? What about the interaction between THC and alcohol? Right? So this is a really important question to ask. And there are hints that there is an interaction between THC and alcohol and driving outcomes that we should research and be interested in.
Okay, well, this comes to the second question from Mary O'Connor, and she asks about fetal alcohol syndrome and cannabinoids and alcohol. And she was talking about a study which found dispensaries were prescribing cannabinoids for nausea in pregnancy, and was wondering about the interactions there.
Right. So I don't know much about the interaction between maternal use of cannabis and alcohol and outcomes in the neonate, but there is quite a bit of work now trying to understand associations between maternal use of cannabis and outcomes. Now, it's a little tricky because when you look at the data and you look at surveys or you look at dispensaries and what they're telling their patients, their consumers, is we don't really know what people are using and how long they're using it for. So for example, now when you can get all these different products, are women smoking cannabis with high amounts of THC? Are they using oral cannabidiol, which is a very different cannabinoid? So there is a growth of research in this area, as well as in studies in animal models to understand what are the impacts of maternal exposure during those important stages of development. At this point, we know that there are associations with preterm birth, low birth weight. There was a recent article from the ABCD study that looked at psychotic-like experiences in children that were born to women who endorsed using cannabis during pregnancy. So there is some data coming out, but it's really hard to have people recall what they were using, how often they were using, what type of cannabis they were using. So there are quite a bit of unknowns that I think over the next 10 years, we're going to learn a lot more about.
Okay, thanks, Ziva. So Kathy's asking about THC is a gateway drug, and we always have this discussion in class. It's an important one, and she's asking specifically about when it's used for pain management.
Oh, interesting. So like, is it
Yeah.
possible that if people are using THC for pain management, they'll move on to opioids?
Possibly, yeah. I
Right.
mean,
So now, usually it's the opposite now. People
Yeah.
are interested, and actually, Stephanie Lake, one of the postdoc students in our laboratory, has looked at this, is that, sometimes people are now substituting their opioids for cannabis and able to reduce their opioid use, and some are even able to eliminate it. Now, those are survey studies, and there has yet to be a randomized controlled trial looking to see, can cannabinoids help facilitate reductions in opioid use in chronic pain patients or eliminate opioid use. And so this is a really important area of research. The gateway, I don't usually think of it as in the opposite direction for chronic pain, but definitely, we think about the gateway hypothesis as, the kids, they get exposed to cannabis, they move on to harder drugs. But I think the upshot is that in animals, if adolescent animals are exposed to a number of substances of abuse, in adulthood, they are going to use other substances of abuse. And thinking about, from an epidemiological perspective, there's a lot of other variables going on with respect to what variables are associated with later use of substances that might be considered riskier, such as opioids and stimulants and things like that. So I don't think people hold much stake in the gateway hypothesis, just because it's a very complicated story and other really important variables that underlie behavior in adulthood.
Okay, I know we're supposed to stop soon, but maybe we can carry on until we're forced to stop.
Yeah, I'm happy. I'm free.
So Carolyn's asking about the how to better really inform patients of the risks of cannabis use, and at different stages during their life. So I think that's really important, that there are different, risks associated at different stages, and how to sort of communicate this.
Right. I mean, and I think this is something that we're trying to do here, especially, focusing on the aging population. And you think about the risks that we know of right now of using cannabis in the aging population. So one risk is, okay, well, if people, aging comes along with probably using other medications. So what are some of the interactions between cannabis use and those other medications? We know that there are pharmacokinetic interactions that might lead to increased plasma levels of certain medications that older populations are taking. So that's really important. Another important area is the fall risk. Dizziness and fall risk is known to be associated with cannabis use in older populations. And some other ones, cognitive impairing effects, its effects on memory, its effects on attention. So we know that there are, a set of risks that we should be focused on in the aging population, a set of risks that we should be focused on in the younger population as well. How do we educate people? I mean, I think that this comes to, Chris has a initiative where he goes to high schools and he brings college students to educate high school kids about the risks of substance use. And so that's a way that we can get real information to students. I mean, the internet is wide open, and people of all ages are accessing all this information on the internet. How credible is it? So if it's coming from us, I feel like, I feel like we're able to deliver credible information. Of course, there are a lot of unknowns, but as long as that's relayed to students, to community members, at least we can share with them what is known, what they should be made aware of, right? How do you read a certificate of analysis? Do they understand, when you get something from a dispensary, do you know that it's regulated? Meaning that, do you know that that product adheres to California state standards for a cannabis product, meaning that it's tested for pesticides, mold, potency? Do they know how to read the bottle? What does it mean that there's 1,000 milligrams of CBD in here? What does it mean that there's 10 milligrams of THC just in one dose? That's really important that people understand. If you're eating a chocolate bar with 10 doses of 10 milligrams of THC, and you don't understand that each little tiny square is 10 milligrams and you eat the whole thing, you're going to be pretty uncomfortable. So these are really important areas that I think we're, here at UCLA, we're working hard to improve with respect to community education and outreach.
Okay, great, thanks. So Nancy's Nancy Lee is asking a bit more about the gender differences and whether women may seek out cannabis for different purposes than men in general.
Yes, this is a good question, especially given that there are now products that are marketed specifically for women. So there's been a lot of interest now, let's say there was interest in helping with PMS symptoms. Now I'm seeing that there's products that are helping with perimenopause and postmenopause. There are other products that are geared towards women. I remember I talked to some company that was working on a CBD infused tampon. So they were really set on understanding how CBD delivered in this method would be really helpful to females. So I definitely think that there's a whole range of issues that can be studied with respect to consumer behavior, epidemiology, and also understanding just really, do males and females differ? The studies that I showed you, the 94-day inpatient study, specifically recruited for males. They only had males. Of course, that shouldn't come as a surprise. It was 1970. But, even now, with these controlled studies that are happening, it's hard to recruit females. And so what we do here is we're really trying hard to always have even numbers of males and females. So for every study drug that we're looking at, we can essentially compare how males and females differ. We're collecting blood on essentially everybody throughout these drug administration studies. So we can look at pharmacokinetic differences. We're tracking where they are in their menstrual cycle phase. All these are really important variables to be able to give us a window into understanding if males and females differ in their response, and also how might they differ.
Great. And next one is from Connor. He asks us, what were the ages of the participants in the unpublished youth and older data related to intoxication?
Right. So that came from our studies, not here, but at Columbia. And so we were recruiting people who were 21 to 45. So that was 22-year-olds, essentially, plus or minus, compared to 43-year-olds. It was a very small sample, but it was controlled with respect to how much cannabis they were using, which is an important aspect because we want to be able to control for potential tolerance effects. So super small sample, but that kind of laid a groundwork for the grant that we're doing now, where we're recruiting 30 people who are emerging adults, 30 people who are middle-aged, 30 people who are older middle-aged, 55 to 65, and really comparing the pharmacodynamic and pharmacokinetic effects of THC when it's smoked or when it's given orally.
Okay, so I'll pick one of Diana's questions because I think we got to stop now. But are kids whose mums use cannabis more prone to cannabis abuse later in life? I don't know.
It's such a, I feel like it's like low-hanging fruit.
I'm sure.
The mental health issue that people usually focus on are psychotic-like symptoms and schizophrenia. I don't know if there's been studies looking at cannabis use disorder in kids born from mothers who were using cannabis. It's a good question.
Yeah, no, I mean, it's complicated by the environment as well, right? So there's a kind of educational influence as well. So, okay, well, I guess that was just, well, I'll ask Diana's last question. She was really asking about the difference between those who start smoking as opposed to popping pills and the differences there, which are clearly in onset and metabolism and everything and highs and stuff like this. So maybe you could just give them a quick overview.
Wait, this was a question about kids born to mothers?
No. This is the second question. The exact question is, given muscle memory, did you find that some people had to smoke to get the effects of versus popping the pills? So a sort of state dependent way of taking the drug, so to speak.
For which endpoint was this? I forget.
I
for
think,
just sex difference.
Yeah, it's just the effects, right? So it could be the intoxicating, it could be the pain, analgesic effects. It could be lots of different effects.
Right, and so if you're just looking at THC, forgetting all the other stuff that's in cannabis, if you're just looking at THC, and you're comparing oral administration, so like the pills versus smoked, you're going to see a bunch of differences, even holding the dose constant. So you're giving 20 milligrams smoked THC, 20 milligrams oral THC. So, one really important point is that, the onset of effect is going to be delayed if given orally. The magnitude of effect, depending on which endpoint you're looking at, is going to be different, smoke versus oral. And, for sex-dependent effects, actually, I didn't show some of our data, but in our females who use cannabis every day, we don't get an analgesic response with them compared to males who smoke every day. We get robust analgesia after smoking cannabis with THC. So sex, is going to play a role here. And I think age might as well. We'll let you know in a couple of years after that grant's done.
Okay, fantastic. Well, thanks, Ziva, for I think a great talk. So
this is great. Thanks everybody for sticking around and for listening to some fast-paced information. So thank you. Please reach out.